Good evening, one and all. A warm welcome to the third day of International Webinar Series BIOSPECTRUM 2021 on the recent trends in biological sciences organized by the Postgraduate and Research Department of Microbiology, Sri Shankara College, Kaladi. Today, we are having the Technical Session 3 led by Dr. Arun Peethambaran, Principal Investigator, Rotor Army Institute of Research, Silver Spring, Maryland. And the topic of today's session is acute and chronic effects of blast exposure. Dr. Arun has completed his master's degree in biochemistry and also his PhD in neuroscience from the University of Kerala. Later, he joined the Uniformed Services University of the Health Sciences, Bethesda, USA, for his postdoctoral research in neurobiology. At present, he is working as the principal investigator, Rotor Army Institute of Research, Silver Spring, Maryland. And now, before we begin, I request all the participants to mute your microphone and turn off your video for the smooth conduct of the session. And I would like to invite Dr. Mohan, Associate Professor, Postgraduate and Research Department of Microbiology, Sri Shankara College, Kaladi. Sir, please.
Okay. Good afternoon, everyone. Dr. Arun was actually 3 years junior to me in the Department of Biochemistry for having his MSc Biochemistry. Ever since early 90s, we are close friends, and our family also very close to each other. We used to visit each other, and one of my close friends, I am happy to have him to the BioSpectrum, and he was having PhD in neuroscience from University of University of Kerala, and he worked for a short period in Penfold, and then he moves to US for postdoctoral research, and then senior scientist, and finally research investigator, principal investigator in Army Institute. And he has 2 patents, and he published more than 70 articles and 3 book chapters. And very publication, when we go through his publications, it is very active in neuroscience research, and especially recently most of them are blast related, so it's a very hot topic, and many of his abstracts and publications gets very high approval from the field of neuroscience. And at present, Dr. Arun is working as principal investigator, Walter Reed Army Institute of Research, Silver Spring, Maryland. Welcome, and the dais is yours, Arun, and everyone, every other participants. Enjoy this session and make it an active participatory session. Thank you. Arun, now it's your turn.
Thank you, Mohan, for the kind introduction, and thanks to the whole organizing committee for inviting me for this talk, and thanks to all those participants who already joined for the talk. First, let me try to share my slides. Can you all see?
Yes, sir.
Yes, yes.
Good. So as Mohan said, for the recent years, I was working on blast-induced traumatic brain injury. So I believe at least some of you are familiar with traumatic brain injury, and that is induced by blast exposure. So blast means the real explosion, which is mostly occurring in the battlefield and also in most of the terrorist scenarios. So today I will be just talking about the acute and chronic effects of blast exposure. So what will happen acutely and what will happen in the chronic stages after the blast exposure. So this is basically a disclaimer for the Walter Army Institute of Research where I'm working. This is under the Department of Defense. So whatever I am telling here is only my views and not the views of the Army or the Department of Defense. So just put some pictures here. So this one is one of the real blast scenario which is occurring in the battlefield. You can see the explosion here, and this also could at the field, but they are actually conducting training. So this also you can see that this is a training scenario, and you can see that they are using heavy weapons. So. And this also in the training, and this at the training, and this also the training scenario. So why I am putting these training pictures here? So here you can see the real explosion, and you know what is actually injurious after an explosion. So you may think that the sharpeners coming out of the blast is the one of them. Yes, but as you know, not all the blast scenarios occurs with these sharpeners. So some of the blast is a clean blast. There is no sharpener, nothing, but it's very injurious to your brain and also the whole body, not only just brain, the whole body. So what is injurious in the blast is the high blast overpressure waves, which is coming out of the blast. So above the atmospheric pressure, anything which is above atmospheric pressure can be injurious to the whole body. But in this case of blast exposure, you will be experiencing very high overpressure, and that will be injurious to the whole body, not only just brain. But here we are talking only about the injury occurring to the brain. And not only at this explosion, when you use heavy weapons, that also can generate blast waves. So even though they are putting hearing protections here during training, so you can see that the brain is not protected, only the ear. They are worried about the ear, but there will be significant amount of pressure generating from these heavy weapons also. So what I'm saying that not only at the field, but during the training process also, they will be exposed to high overpressure waves, which can repeated exposure to these high overpressure waves can also be injurious to the whole body. So this slide shows that the Department of Defense, USA, they have a National Defense Authorization Act, which came out in 2018. And sorry. And why I am showing this slide is that they are saying that longitudinal medical study, that is long-term study on blast pressure exposure of members of armed forces is absolutely necessary. The question is that why it became necessary. So in the military, what is happening is that after these soldiers exposed to blast, they immediately generate neurological problems because the brain is injured. But so far, there is no real treatment against blast-induced traumatic brain injury. So when the soldiers develop this kind of neurological problems, they will be treated only symptomatically. So what is happening is that after this symptomatic treatment, the soldiers feel better, and after few weeks or few months, they usually go back to the field again, the battlefield again. But what is happening is that after several years, while they are in the military or when they are retired, several years later, they started developing severe neurological problems. And such the secondary effect of that blast exposure has not been well studied, and that is the reason why this National Defense Act saying that a longitudinal study is necessary to find out the secondary pathological mechanism leading to these long-term effects of blast exposure. So the major research gaps in blast TBA is the mechanism of injury is unknown, as I said, and so far no effective therapeutic strategies for prevention or treatment. And no preclinical models have evaluated both acute and chronic effects of blast exposure. So only the acute effects of blast exposure has been studied well, and not the chronic effects. So it is absolutely necessary to develop animal models which can be used for studying the chronic effects of blast exposure. So we developed one animal model, and the animal model we use, we use these male Sprague-Dawley rats, and this is an advanced blast simulator, the machine which used to simulate blast in the lab. So we cannot do the real explosion in the lab, as you know. So we created a machine and which can generate blast over pressure inside this machine. And I will give more details in the next slide about this advanced blast simulator. So using this advanced blast simulator, we exposed these Sprague-Dawley rats to a single blast or repeated blast. So in the field, it can be exposed to a single blast or repeated blast, right? Not just single blast. So there will be boom, boom, boom like that. The blast can occur. So for the repeated blast, what we did was we exposed the rats to 2 blasts within 2 minutes. So there is a single blast and also a double blast which occurred in 2 minutes. And the pressure we used was 19 psi above the atmospheric pressure. So very for the animal, it can be injurious to with the 19 psi. So even human beings can be very injurious with this 19 psi blast. So the preclinical studies and also clinical studies already shown that if you expose to a pressure of around 4 psi, your hearing function, that is auditory function, can be affected. Your vision can be affected. And if you expose to repeated blast at 4 psi, you will have neurological problems too. So even a 4 psi is kind of injurious. So you can imagine what will happen at 19 psi. But as you know, this compared to humans, these animals are very strong. Like they can withstand most of the things, and also they will recover very fast compared to human. So if we expose to a blast like 19 psi, it will take several months for us to return back to the normal. But these animals come back after exposure, they will come back to normal the next day. So after the anesthesia, you can see that these animals are moving almost same like the other sham animals. But we exposed to this kind of a high pressure waves, we will be in trouble. So after the blast exposure, we did neurobehavioral tests, different neurobehavioral functional tests from day 1 to 1 year. So that is a long time for animals. So we can see that a approximate comparison, 1 month of a rat correspond to 2.5 years of human life. And in that 6 months correspond to 15 years. And 1 year corresponds to almost 30 years of a human life. So when we do the assessment from day 1 to 1 year, we are basically comparing the human life from day 1 to 30 years after the blast exposure. So just a approximate comparison. So this is the advanced blast simulator I was talking about. So just go through the briefly how we generate the overpressure here. So there is a driver section here. So we use compressed air to generate pressure in this section, okay? And there is transition section here, and these two sections can separate with a, you can see a gap here. We fill the gap with a membrane, can be fabric membrane or a different kind of membrane. So we use fabric membrane. So these two sections separated by a membrane, and when you generate pressure this area, so with the pressure this fabric membrane will blast or break, and that generate the pressure, and it goes here, and the animal will be kept here. Animal will be looking at this towards this area, towards the driver section. So animal is sitting here, but they will be under anesthesia, but their face will be facing to the driver section. So the pressure will be coming this way and hitting the animal sitting here. And there is a dump tank here, and what it is doing that the waves, the pressure waves coming here after passing the animal comes here, and the dump tank will eliminate the pressure waves. That is the purpose of this one. Otherwise, this pressure waves will come back and hit the animal again. So in the real battlefield conditions, it is plain field, right? So there is no reflective waves. But if it is happening, if the soldier is actually standing near a wall, brick wall, then yes, the soldiers can get reflective waves like here. But we wanted to avoid that reflective waves, so we put a dump tank here that eliminate the reflective waves coming back to the animal. So the animal will be exposed to the blast under isoflurane anesthesia. So animal doesn't know when it is getting the blast. So when they recover from the anesthesia, then they may feel some pain about regarding the exposure. So I will try to show a video here. Hope everybody can see. So what is happening to the animal inside the chamber when you expose to blast. So the video I'm showing here is around 20 seconds, but it is the video was taken using a high-speed video camera, and basically the whole thing is happening in 5 milliseconds. So even though I'm showing it in 20 seconds, it is happening basically in 5 milliseconds. So 1 second is at 1000 milliseconds. So 5 milliseconds, you can imagine how fast it is. But here it is showing it in 20 seconds. So you can watch that video, what is happening to the animal inside the blast chamber. Oh, did it work? So this whole thing happened in 5 milliseconds, but you watched in 20 seconds. So animal is under anesthesia, is secured with a wire net so that the animal cannot move during the blast exposure time. So you see that how it is happening to the animal. So when this real blast happens, this is what occurs in the human body also. See, there is a slight movement of the head also here, which you won't feel when you really see the exposure. You are seeing here because it is slow motion video. So we did one study using this BLAST model and then published last year in Journal of Neurotrauma, and the title was Long-Term Effects of Blast Exposure, Functional Study in Rats Using Advanced Blast Simulator. So in this paper, we reported only the neurobehavioral functions after blast exposure. So I just go over the data with, in that paper. So rotating pole test, that is one of the functional tests we use for the study. So it is a measure of vestibulomotor function. I don't know how many people here in the audience familiar with the neurobehavioral functional test, and I am almost sure that nobody have studied this particular test, but this is very rarely being used. I will show you a next test, which is Rotor test, and that is widely used in the neuroscience field. But rotating pole test is not that widely used. So what we are doing it here is that you see the animal sitting here, and this is a rod, and the animal will be moving from this end to this end. But for the animal, it is easy to move through the rod. But here what we have done is we rotated this pole. So you can see that clockwise, when the animal is moving from here to here, the rod is rotating clockwise. So the animal need a lot of concentration, balance, and coordination because the pole is rotating. So if it is a stable rod for the animal, it is okay. But when it is rotating, it will be hard for the animal to move from this end to this end. And there is a dark chamber here that is attraction for the animal because the animal likes to go to the dark area all the time, not the light, not the place with the light. So animal likes to move there, but it will be hard for the animal to move because it is rotating. And when the animal is injured, at that time it will be more difficult because they will lose the coordination. So here I put the data. The green one is sham animal. Sham means no injury. So only the animal was anesthetized, but not exposed to blast. And the blue one is the animals exposed to single blast, and the red one is the animals exposed to double blast. So you can see that the single blast did not make a big difference in terms of statistical significance, but the repeated blast, you can see that the animals lose coordination and balance, and they can see that they are falling from the rotating pole within the whole test is for 3 minutes, but even though the whole test is for 3 minutes, the animal falls before the 3-minute time frame. The performance score is based on how well they moved, whether there was any fall, how much time they took from this end to this end. So everything count and make a performance score for the animal. You can see the repeated blast exposed animal, the score was less day 1, 6, 14, 1 month. And you can see that after 1 month, there is some recovery. And then after 6 months, they are going back to the same board again, the repeated blast exposed animal. So you may ask why the animals were tested only 6 months, not 1 year. The reason is that these animals, after 6 months, what we found that they don't want to do the test. So immediately when we put the animals here, they have started falling. They don't want to do the test because maybe they are old and also they develop some weight with the time, right? So we try to reduce that one, the weight effect, because we try to diet restrict these animals so that the animals won't gain too much weight. But even then they develop some weight, and also when they get old, they may think that anyway, I'm going to fall in between, so I will fall on the right spot, the beginning itself. So they did not do the test after the 6 months time. That's why I did not put the data here. Same thing for the next test, that is a rotor balance test. You can see the data is only up to 6 months. So in the rotor rod, this is a rotating rod, and the animal, you can see that the animals are hanging there, So while it is rotating, the animals has to stay on the rod. So this also for 3 minutes, but the animal with the time they cannot hold on it, especially if they are injured, they will fall. And when they fall here, the machine will record when they fell. And you can see that here also the green one is for sham, then blue for single blast, and the repeated blast here. So the repeated blast exposed animals, they are falling way early compared to the other two. And you can see these animals, the repeated blast exposed animals is almost the time they are spending on the rotor is almost zero. That means they are not even doing the test. They are immediately falling. And you can see all these animals, including the sham animals, after 6 months, are almost back to the zero time. That is, they are not doing the real test. So we stop the test here. But the take-home message is that the repeated blast exposed animals, their balance function and coordination, they are all losing with the time and significantly. So the next test we use is novel object recognition test, and this test is basically used for measuring their short-term memory. So what we will do is that we put the animal in a box with two identical objects. You can see that two objects are similar, right? And we let the animal to explore these two objects for 3 minutes. And then we take out the animal, keep this box, and the next one after 5 minutes, they will come back to the box. When they come back, we replace one of the old object with a new object. So you will see, you can see a different color, right? So a different object here. So if the animal is having memory, the short-term memory, then the animal will go to the new objects most of the time, because the animals, they wanted to explore new objects all the time. So if they find this one and if they have the memory, they don't care about this one. But if they find a new object, they will spend more time with the object, exploring that new objects. So we calculate a discrimination index using the time they spend with the novel object and familiar object and the total time spent. So you can see that this is the sham, that is uninjured animals with a different time, day 8, 1 month, 3 months, and 6 months. You can see that the blast exposed animals, the discrimination index is significantly low. So not only just double blast, here the single blast exposed animals also behaving like crazy. They don't have they are not showing a ability for the short-term memory. And this test also we did finish by 6 months, because they are huge and they were not interested in doing the test again here. The next test we used was Morris water maze test, and this test is used for long-term memory functional. So you can see the animal is here. This is a tank filled with water. The animal will be allowed to swim in the water, and you know that they don't want to swim and they don't like to swim, and they will be scared if they are put in the water. So they want to escape. So we put a we put a platform here, and the animal will look into these visual cues. You can see the different shapes, and they find the platform, and then they know that the platform is near this object. So they will have the memory next time after the training, saying that, oh yeah, the platform is here near this object. So if they have the memory, they will easily find the platform where it is. But if they lose the memory, we do the test next day after the training. If they lose the memory, they will take long time to find the platform in the tank. So You can see that the test results show that at 1 week, the sham animals here, single blast here, and then the repeated blast here. So the blast exposed animals are taking more time to find the platform because they lost the memory. After the 1 week, you can see that there is no statistically significant differences except at 9 months. So after some time, the secondary effects is occurring, and then you can see that 9 months there is significant difference here, especially in the single blast exposed animals. And we did not see much significant changes at 1 year when we did the test in 1 year for this particular one. The next test we used was open field exploratory activity test. So this is a box, and you leave the animal inside the box for 1 hour. The animal will be allowed to explore the whole box for 1 hour. So if the animal is normal, the animal will go all the places, come to the center, go to the corners, and move all around this chamber. But if the animal is having anxiety or depressive like behavior, they usually won't move much. Mostly they will stay in the corners on the edges of this arena. So we measure different parameters like total distance moved. That is the total distance the animal moved inside this box within the 1 hour time period. So you can see that the sham, single blast, and double blast, the double blast exposed animal shows significant difference at day 1. That is, they are moving less distance inside the chamber. And you can see that only on day 6, no significant change. 14 days still only the double blast exposed animals showing changes, and they are back towards normal again by 1 month. But you can see that after 1 month, the secondary effects you can observe, and then you can see that significant decrease again at 9 months, and also you can see a trend towards decrease movement after 3 months, 6 months time frame. And 12 months also you can see the different, but statistically not significant. That means that the blast exposed animals, especially the double blast exposed animals, are moving very less inside the chamber. So center time legacy. So as I said, if the animal is having depressive like behavior and all, the animal will stay mostly at the corners or the edges. They won't come much to the center of the arena. So this particular measurement, this one measures how much time they usually come to the center or cross the center of the arena. So you can see that the blast exposed animals, especially the double blast, you can see that they are coming less to the center, less towards the center, and that stays there for 6 days, 14 days, and 28 days. You can see that single blast exposed animals recover, even though statistically not significant. You can see a decrease here, but they recover after some time. But even the single blast exposed animals, you can see that start coming down after some time, especially at 6 months and 9 months. You can see even the single blast exposed animals are showing that it kind of anxious and depressive like behavior. This is the last one for the neurobehavioral functional test. This is the margin time legacy. That means how much time the animals staying on the margin. So if they are not coming towards the center, then they will be spending more time right at the corner. That's what we are seeing. The previous one, center time was less. That means here the margin time will be higher. So you will see the same changes, but should be on the opposite side. You can see that double blast exposed animals significantly different. So single blast is there, but statistically not significant. But even the single blast exposed animals, you can see statistically significant difference after 6 months. So the secondary effects occurs after 6 months, basically. So the study confirmed that blast exposed animals they lose neurobehavioral functional ability with the time, and acutely they show significant changes and they recover after 1 month, but significant changes occurs again after 6 months. So there is acute and chronic changes after the blast exposure. So most of the previous studies, as I mentioned, studied only the acute effects. So the study is showing that there is chronic effects which is occurring in these rats after 6 months, basically statistically significant difference. So this can be a good model for studying the acute and chronic effects of blast exposure. So here I'm putting two different publications from a different group. Yeah, before going to this publication, anybody has any question regarding the previous study, the neurobehavioral functions? Okay, if there is no questions for the previous study, so I will go to the next one. So this one, these two studies published same time in 2015, but two different groups, but both their conclusions were similar. So what they have done is that they've studied soldiers who came from the Iraq war, the Gulf War. So they choose soldiers who did not expose to any kind of brain injury. They also selected separately those soldiers having brain injury, but not because of blast. And also they selected those soldiers who had brain injury, and that was due to blast exposure. So, so control groups, no injury, and then TBI with different forms of TBI, and also third group was those having brain injury due to blast. And both their conclusion, they did diffusion tensor imaging technique to find out what is happening in the brain in those soldiers who were exposed to blast. What their conclusion, both study groups, their conclusion was the soldiers who exposed to blast, their myelin integrity is decreasing way fast than the controls or those exposed to different kind of brain injury. In other words, so as you know, when we are all getting age, our myelin integrity, the brain myelin integrity started losing. But according to this study, what is happening is that those soldiers who are exposed to blast, their myelin integrity is getting disrupted way earlier. Or in other words, the soldiers who are exposed to blast, they are getting aged faster than the others. So you will be aged faster than the others if you exposed to blast. That is the conclusion basically. So we decided to study this one in our animal model. So as you know, we cannot, we don't have this diffusion tensor imaging machine in our facility. So we decided to do this one biochemically and pathologically instead of doing imaging technique. And we did one study to find out whether blast exposure is accelerating the brain aging process. And the data we published last year in Frontiers, and what we found that blast exposure leads to accelerated cellular senescence. Senescence means the aging process. So the cellular senescence is increased after blast exposure. So I will show you how we did the study. So we use the same animal model which I explained previously, and we use 3 different markers for cellular senescence or aging. The first one is senescence associated beta galactosidase enzyme activity using histopathological technique. So you may know beta galactosidase enzyme for sure. But this senescence-associated beta-galactosidase is a modified form of this beta-galactosidase, but this enzyme is expressed only in those cells which are undergoing aging process or senescence process. So normal cells, they don't express this senescence-associated beta-galactosidase. Only the aging cells express this enzyme. And this enzyme is very stable also. Even this enzyme is stable above 55 degrees. So it is different than the normal beta-galactosidase enzyme. We also did measure the expression of senescence marker protein 30, SMP30. So this protein is also well studied as a marker of cellular senescence. The expression of this protein, SMP30, decreases with aging. So when you get aged, the expression of this protein decreases. And P21 is another protein, and the expression of this protein increases with aging process. So opposite effect of these two. So this one decreases and this one increases. So senescence-associated beta-galactosidase activity we measured using histopathology. So it's a staining process. So here we can see that sham animals, single blast and repeated blast, 24-hour, 1 month, and 1 year. You can see that, and this is the densitometric analysis of this staining. So if you look into the densitometric analysis, you can see that the 24-hour itself, you can see the statistically significant increase the number of cells undergoing senescence or the aging. So immediately after blast exposure itself, the aging process starts. And 1 month, you can see the more obvious changes. You can see the significant difference. But 1 year, we are not seeing a statistically significant change, but you can see a trend there. But you may ask why even in the sham, there is an increase with the time that is expected, but we are not seeing an increase here after 1 year compared to 1 month. So one reason we are thinking that these cells undergoing senescence, they will be taken up by the immune cell that is already reported. So once they're taken up by the taken up and removed, then you won't see much staining here. Another reason is that we were using different batches of kit from the company for each time period. So as you know, the different batches of kits can also make some difference. So the difference here could be due to two different reasons. One could be the different batches of kits used, and also the cells undergoing senescence will be removed by the immune system in the brain. And this staining was done in the motor cortex region. And we will I'll show you a few other brain regions. We did the ventral thalamic nucleus region, including which is involved in moving function. Here you can see that the changes are same almost. You can see immediately after blast, that is 24 hours, you can see the significant difference. And there's a huge difference you can see in this at 1 month, especially the repeated blast exposed animal. And the statistically significant difference is still there at 1 year. This is another brain region, auditory cortex. This is involved in hearing function. So the brain region involved in one of the brain regions involved in hearing function is auditory cortex. Not much changes at 24 hours, but statistically significant difference at 1 month. Not much at 1 year, but you can see a trend there. Dorsolateral thalamus, another region involved in ocular function, and it's almost same thing. Increase here 24 hours, 1 month, and 1 year. Statistically significant difference at 1 month. Superior colliculus, a brain region involved in the ocular function. We can see all the time points we are seeing increases, but statistically significant difference at 1 month. And then next region is the geniculate nucleus. Here also you can see changes at 24 hours, 1 month, and 1 year. So This brain region is important, hippocampus, and this is the brain region involved in memory function. And you can see that statistically significant difference at 1 month and 1 year. So the cells in the brain region which is involved in memory function, they are getting old after blast exposure. So then we did QRT-PCR analysis to determine the expression, differential expression of the SMB30 protein and P21. As I said, the expression of SMB30 decreases with age and P21 increases. So that's what we are seeing in the test also. The blast exposed animals, especially repeated blast exposed animals, you can see that statistically significant decrease in the expression of SMB30 and increased expression of P21, which is support the previous one of SA beta galactosidase enzyme activity. So all three are showing that blast exposed animals, there is acceleration of brain aging process, which support the previous clinical finding in human patients, saying that blast exposed victims, their brain is getting aged faster than the controls, or those did not expose to blast. Any questions from this study? Or you can ask the question at the end. I'm going to talk only one more data from this study. That is blast exposure, acute and chronic auditory dysfunction. So auditory dysfunction means the hearing function. So what is happening at acute and chronic stages after blast exposure to the hearing function? So in this study, we did not measure any auditory functional test. We did one pathological test only here, and that is neuronal degeneration, the extent of neuronal degeneration using silver staining. So silver staining is a staining technique which measure the extent of neuronal degeneration. So when we are all getting age, there is degeneration of neurons. So, and if there is an injury, then these neurons can undergo the degenerative process faster. And that can be measured using silver staining. So we use the same animal model and we perfused the animals, collected the brain regions, and did the silver staining. And the data we just published very recently in March in Frontiers again. So what the conclusion is blast exposure causes long-term degeneration of neuronal cytoskeletal elements in the cochlear nucleus. Cochlear nucleus is the brain regions which is mainly involved in the processing of auditory functional signals. So I will show the first slide, only 2 slides in this data. So sorry for this is a very busy slide, hard to understand, but there is not much information in this slide. That's why I put all the 4 figures together. So these are the different brain regions involved in hearing function: auditory cortex, medial geniculate nucleus, lateral lemniscus, and inferior colliculus. So all these brain regions are involved in the hearing function. The auditory cortex is here, medial geniculate is over here, and the lateral lemniscus is all the way here, and the inferior colliculus is over here. So we did do the silver staining in the brain sections at 24 hours. This is 14 days, and this is 1 month, 6 months, and 1 year. And this is on the left side, and this is on the right side. So those 2 sides we did separately. Same here also. So different time points and 2 different sides. So the conclusion from this particular slide is that we did not see any significant change in the neuronal degeneration in any of the 4 brain regions. So all those 4 regions, even after blast exposure, we did not see any change. after single blast or repeated blast. Next brain region we studied was the cochlear nucleus, and this is the cochlear nucleus, left side and right side, and this is the cerebellum. You know that this is the cerebellum of the brain, and this is the brainstem area. So it is over here, the cochlear nucleus, and in the animal, the ear is all the way here. The ear comes over here, and as you know, then cochlear nucleus is the immediate brain region close to the ear. So the first brain region which is involved in processing auditory signals, which is coming from the ear. So when you hear those signals come here and process here first, and then go to the other brain regions which I described in the previous slide. So when we did the silver staining in the cochlear nucleus, we saw significant changes. So this is 24-hour, 14 days, 1 month, 6 months, and 1 year. And this is the left side, and this is the right side. So you can see that staining, the sham it is less, but you can see an increased staining in the blast exposed animals. And you can see the same thing here and there. So on both the left side as well as on the right side, after single blast and repeated blast, we are seeing a significant increase in neuronal degeneration. That is, the neurons in that area is undergoing neuronal degeneration, or those cells undergoing degeneration will later will be non-functional, and then they will die. So these two figures showing the densitometric analysis. The black one for sham, then single blast, and then repeated blast. So compared to the sham animals, you can see all the time points we are seeing a significant increase, except maybe 1 month, statistically not significant, but there is a trend there. This is for the left side, and this is for the right side. You can see all the time points in the right side, you can see statistically significant increase in the neuronal degeneration, and no statistically significant difference between single and repeated blast. So just a single blast was good enough to simulate this neuronal degeneration process in the cochlear nucleus. So since this region is close to the ear, so what our conclusion or hypothesis is that the blast waves, blast overpressure waves are coming through the ear canal, and then it actually, since it is 19 psi, it will rupture the tympanic membrane. So even pressure above 4 psi, if it is 4.5 psi, itself can rupture the tympanic membrane. Here we are using 19 psi, right? So it is very high. So it will definitely rupture the tympanic membrane and maybe directly impacting the cochlear nucleus. That's why we are seeing neuronal degeneration in this area and not seeing neuronal degeneration in the other brain regions involved in auditory function. So we propose that if you put a good hearing protection device, it can prevent this from happening. So if you put a good hearing protection device, it can prevent the entry of this blast or pressure waves and injuring the cochlear nucleus that is in the brain. And so the soldiers in the US military, so now the main problem is that the soldiers are having difficulty in hearing, and also most of the soldiers, they have tinnitus. I believe some of you know tinnitus. Tinnitus is ringing in the ear. So you will feel that you are in the ear, you always hear a ringing like of noise. So that is called tinnitus. So the US military, a lot of soldiers are suffering from tinnitus, and this brain region is involved in causing this tinnitus problem. So if the cochlear nucleus is injured, then there is chances of tinnitus. So this explains why the soldiers are having auditory functional deficits and also developing tinnitus. So the take-home message is that this the rat model we have can be used for studying both acute and chronic effects of blast exposure. So the conclusions here are our rat model of blast TBA using the advanced blast stimulator can be used as a model to study acute and chronic effects of blast exposure. And blast exposure causes acute and chronic changes in the brain leading to neurobehavioral deficits. Blast exposure accelerates cellular senescence or aging process. And blast exposure causes acute and chronic degeneration of neurons in the cochlear nucleus, which may be responsible for the auditory dysfunctions, including tinnitus after blast exposure. And our ongoing study using this model is that development of biomarkers of blast-induced TBA and also determine other acute and chronic neuropathological changes after blast exposure. So the funding for the study came from Military Operational Medicine Research Program. They call it as JPC5 here. And these are the investigators involved in this study. They are all helping us in different form. And administrative staffs who helped executing this project. And open for questions.
Thank you, sir, for your informative presentation. And now it's the time for queries, and I request the participants to post your questions in the chat box.
Okay, wonderful presentation, Dr. Arun. One question: is there
Yes.
any medication available for reducing the damage?
As I said, so far, no therapeutic strategies have developed, no drugs, no effective drugs developed for blast-induced TBA. What they are doing is that symptomatically treat them. If they're having some kind of sleeping problem, then treat for the sleep. If they have some other pain, like memory problem, they treat even for Alzheimer's disease. No treatment so far, right? Like that, they treat only symptomatically even for Alzheimer's disease. Same kind of treatment they provide to these patients. And if there is any headache, they give pain medication for the headache. That's it.
Is it possible to have some?
Yeah, that is our ultimate goal. So once we understand the mechanism, so here now we have the model, right, to study the acute and chronic effects. So if you test different therapeutics, and you can even find out the mechanism of the secondary injury using this model, and then treat or prevent the secondary injury also. So that is a long-term effect. That is the main problem. So we are in the process of developing strategies for the treatment. Currently, we are testing multiple drugs. And one drug which we are currently testing, which is just approved for funding, is that, this one, toll-like receptors. So there are so many different types of toll-like receptors, right? One company developed a drug which can actually inhibit toll-like receptors, and that is toll-like receptor activation is one of the immediate pathological changes occurring in the brain after blast exposure and also after other kind of TBA. So if you inhibit toll-like receptors immediately after blast exposure, could be a good therapeutic strategy. That's the one we are going to test it. Like that, people have explored using different other drugs also. N-acetylcysteine is another drug people tested not only in animal model, but even in human, they studied the effect of N-acetylcysteine and found to be somewhat effective, but not completely. But the soldiers actually felt better after treating with N-acetylcysteine, but they could not completely recover from the injury. Progesterone is another drug people tested previously in TBA models, progesterone.
Okay, thank you. And you said we can, the soldiers can be supplied with ear caps or some protections, but I think in the battlefield they should be so vigilant. They need to hear all the small sounds and everything too. Is it possible to have some ear caps during wartime or battlefield?
Yeah, that's a good point. The problem is that yes, they were given this ear. You can see that even in this picture. Let me show you. You see here the green color. I don't know whether you can clearly see. There is a green color that they put it inside the ear. There is a ear plug there. But usually these soldiers are given ear protection devices. But even those ear protection devices, they cannot provide complete protection. This blast overpressure can even penetrate those ear protection devices. If it is high blast overpressure waves, then it can still injure your tympanic membrane and the inner ear. And goggles, and these goggles they were given, but sometimes they don't even put it because they cannot see properly. Same here also, hearing protection, they cannot hear the command from the commander properly, so they try to remove it, and maybe at that time the explosion occurs.
Yeah, it's hard for them to wear all these things.
Yes. Wow. And also they are all heavy. Even this helmet, they are all heavy. They protect from blast, but not completely. And you will see that this one, these are pressure sensors. So in the battlefield, the helmet they are given will have the sensors also to measure how much blast overpressure they experienced, and they will be treated if they get too much blast overpressure, which will be obtained from the sensor value.
Okay, thank you.
Okay, sir. There are some questions in the chat box also.
Okay, let me go there.
Okay.
First question I can see is there any significance for using 19 psi as blast? Let me see. That is the first question. Oh, so many other questions. So from where I should go? From bottom to top or top to bottom? Okay, let me go from the
Your
top.
wish, sir. Okay, sir.
Okay, it says which type of ear aid must be used while explosion. So there are different kinds of ear protection devices, and still they are developing more. So these are all secret information. So they don't share that much information, and also we don't know exactly what the real type of hearing aid they are given. But there are multiple ear protection devices available in the military. So they may use the best one they like it. The next question is, is there any significance for using 19 psi as blast pressure in your study? The main reason is that this is the highest pressure we can go in that machine. So we try to expose the animal to the highest blast overpressure. So in that machine, we cannot go above 19 psi. So that was the reason why we use 19 psi. We can go below, but
Arun, you're muted. Unmute here.
Okay. Did you hear the answer for the previous one?
The previous we heard.
Even the 19 psi, did you hear?
Half we heard.
Okay. The question is that why I use 19 psi blast. Okay, the reason is that using the advanced blast simulator, we can go to the maximum of 19 psi only. So that is the reason why we use 19 psi. So we can go below 19 psi. That's not a problem. We can change the membrane, but with the maximum thickness membrane which we use, we can go only up to 19 psi. So the highest pressure. That was the only reason choosing 19 psi. The next one is that have you studied myelin integrity in Alzheimer's patients? If these blast exposed soldiers are more prone to Alzheimer's disease. Yeah, we did not study this one in myelin patients, and I don't know anybody studied in Alzheimer's patients. This myelin integrity should have done, but since we are not interested in Alzheimer's disease, we did not explore. But currently, the hypothesis is that those exposed to blast, they are more prone to Alzheimer's disease. There are so many recent publications came and also some reviews. They're all suggesting that if you exposed to blast, you are prone to develop Alzheimer's disease. But we don't have much data. One data which we had earlier was we were checking the expression of phosphorylated tau. You know, tau protein, which is the cytoskeletal protein, the neuronal cytoskeletal protein. So the normal tau is good for the integrity of neurons. But once this tau gets phosphorylated, which actually causes degeneration of neuron. So what we found that after blast exposure, this phosphorylation of tau occurs. So, and this Alzheimer's patients also, or in Alzheimer's models, we already people already reported that this phosphorylation of tau occurs, hyperphosphorylation of tau. So we also think that that could be one thing which actually can trigger Alzheimer's like pathology after blast exposure. So tauopathy, the tauopathy is well known in Alzheimer's disease. And we already saw that there is phosphorylation of tau occurs after blast exposure. So we also think that yes, blast exposure can trigger Alzheimer's pathology, but we did not explore that one. But with using this animal model, we can definitely go for, because we are studying the effect till 1 year, right? Okay, next question is blast-induced cellular senescence can be controlled by administration of anti-cellular, anti-aging components. Yes, yeah, it is possible. Yeah, there are anti-aging medications that are available, and also um, 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 there is inflammation is associated with this cellular senescence. So anti-inflammatory drugs can also be found to be effective, but we did not study that further. We just only observed that cellular senescence, acceleration of cellular senescence occurred after blast exposure. So yeah, those therapeutic strategies should be explored. Next question. I did not see anything in this chat. Let me see. Let me go up.
Do you have any other questions?
Yeah, I did not see anything else other than these.
Okay, and I think there is no more queries. Thank you, sir, for answering the questions. And now I welcome Parvati Sushilan to deliver the vote of thanks. Parvati, please.
Am I audible?
Yeah.
Yes, yes.
Good evening, one and all. On behalf of Postgraduate and Research Department of Microbiology, Sri Shankara College, I take this opportunity to extend my sincere gratitude to our resource person, Dr. Arun Peethambaran, for sharing your excellent lecture. The section was informative and enlightening. Thank you, sir, for spending some of your valuable time with us. I also thank all the participants for your cooperation and active participation throughout the section. Once again, thank you all.
Thank you.